We're gonna properly start with our with our MicroVision Talks today, career path development. Just for reference, this is an online seminar series which is hosted by the Puerto Rico Society of Microbiologists that is designed to guide microbiology professionals through various career opportunities, offering expert advice, networking tips, and strategies for success in the field. For today, we are proud to have Dr. Anna F. Law, uh, presenting us a presentation titled Preparing for an FDA Inspection. Myself, I am Dr. Eduardo Tosado Rodriguez. I am part of the board from the Puerto Rico Society of Microbiologists, and it's an honor to be presented here today. I'm gonna be reading a short bio about our speaker, so you can have an idea of her background, and you can develop questions if you're interested in the topic. So today we're honored to welcome Dr. Anna F. Law, a nationally recognized clinical microbiologist and an expert in sterility testing and rapid microbial detection. Dr. Lau received her Ph.D. from the University of Sydney, Australia, where her research focused on the development of novel diagnostic platforms for invasive fungal diseases. She completed her CPIP Clinical Microbiology Fellowship training at the NIH and is board certified through the American Board of Medical Microbiology. In 2013, she joined the Clinical Microbiology Service at the NIH Clinical Center, where she co-directed the bacteriology, specimen processing, parasitology, and molecular epidemiology sections. In 2018, Dr. Law was promoted to chief in the newly created sterility testing service to support the NIH CGMP program for cell therapy and drug pharmaceutical manufacturing. There, she leads the development and validation of microbiological methods that support advanced clinical, cellular, and gene therapy programs. Dr. Law champions the education of clinical and pharmaceutical colleagues for GMP testing within a hospital environment. Her current research involved the development and evaluation of rapid microbial test methods for sterility testing and mycoplasma testing. Dr. Law serves as the 2025 until 2030 USP Microbiology Expert Committee, PDA Microbiology Organizing Committee, editorial board for the journal Clinical Microbiology, and is an associate editor for Frontiers in Cellular and Infection Microbiology. She is a gifted educator and communicator. Dr. Law is an ASM distinguished lecturer and a frequent invited speaker at the major scientific and regulatory forums. Without further ado, please join me in welcoming Dr. Anne F. Law for today's presentation.
Thank you, Eduardo. Hi, everyone. Let me start sharing my screen. And if someone can just confirm that you can see the slide deck in the right format.
Yes, we can see the slide deck perfectly.
Wonderful. Awesome. All right. Hi, everyone. Thank you so much to the organizers, Natalie in particular, Eduardo, for inviting me to present. I'm excited to be here, and thank you to you for all the time that you're taking out to hear the seminar. I understood that it was a career path development, and so I tried to build within this presentation a little bit of a story and a little bit of a weave to give you guys a little bit of a taste of, I guess, my background and where we're coming at when we're talking about preparing for an FDA inspection. So as Eduardo mentioned, I've been invited through the ASM Distinguished Lecturer Program, which has been running for 60 years and has presented over 1,000 lectures. It is provided by ASM specifically for the various ASM branches and is funded through ASM. ASM is fantastic. I encourage you, if you're not a member yet, to please consider joining up for your membership. It gives you access to cutting edge science, networking, career advice, etc. And then the membership is open now for 2026 as well. All right. So I don't have any financial disclosures, and everything that I present today is all my opinions and does not reflect the NIH or the US Department of Health and Human Services. So what I plan to do today is, as I said, walk you through our story, or walk you through my story, which is really starting at an intersection of health microbiology that leads to an event that happened at the NIH in 2015, and I think it's very poignant to point that out. My understanding is that the audience here today may be from diverse backgrounds, and hopefully some of this story you'll be able to connect with. Then we'll move into the FDA inspection process and preparing for an FDA inspection. Okay, so the intersection of health microbiology. When I think of health microbiology, they fall into two different fields, with the yellow one being clinical microbiology, the blue one being pharmaceutical microbiology, and then we've got this kind of a gray-green zone in the middle. So for those of you who are in the clin micro field, this would be very familiar to you. For those who are not in the clin micro, essentially this is diagnostic testing. It's clinical pathology, it's infectious diseases. When you're sick, you go to the physician, you may submit a sample. That sample then goes to the clinical pathology lab or the clin micro lab where they process it. They'll either do molecular testing to find out what microbe it is, or they'll do culture testing followed by susceptibility testing, antimicrobial resistance testing, and it's all about rapid diagnostics in this particular field. We then have the other branch of health microbiology, which is covered under the pharmaceutical arm, and this now has a very different scope. We are now focusing on product quality, so think about the various drugs that we may all be exposed to, whether they're allergy medication, high blood pressure medication, etc., or if you may be going to the hospital, saline drips, antimicrobials, etc. They're all products and fall under the drug category. So we care about the quality, we care about consumer safety. And here, the big thing here is microbial contamination control. So you want to make sure that whatever product is going into patients is either sterile, if it's meant to be a sterile product, or it's at a sufficient level of acceptable bioburden. And this falls under the current good manufacturing practices, which is a regulation under the FDA. So again, just to give you some different aspects of what I just described, in clinical micro, we're thinking about syndromic panels. And some of you may be familiar with, the BioFire system, the Cepheid system. We think about those clinical micro labs being accredited, usually via the College of American Pathologists. We also think of clinical micro labs being able to respond rapidly to emerging events, such as the COVID-19 epidemic, where essentially laboratory developed tests need to be set up in-house for diagnostic testing. On the other side, on the pharmaceutical side, we're thinking about products and safety. And so what I put here are 3 examples of what has been recently on the lay news, right? So you may recall on the news that there was fungal contamination in cysteine eye drops. This was in December 2024. That led to recalls, and it led to several deaths in patients that were exposed to these contaminated products. More recently, there is investigations with regards to infant formula and the fact that there's Clostridium botulinum present in these. And then recently this year, we have a drug tablet. So trimsulfa, which is an antimicrobial, was recalled as well because of microbial contamination. So often these things are happening in the background as regulated by the FDA, and we thank our FDA regulators for providing safety oversight of that. But obviously when things hit the news, that's when they're major. And you may also recall back in 2012, there was a nationwide fungal meningitis outbreak due to improper manufacturing and compounding of drugs, of basically a steroid medication that was being injected into the spinal cord of patients across the US, and over 700 patients were affected and over 60 patients died from that drug product. So this matters because they fall under different regulatory landscapes. So in the clinical diagnostic lab, those of you in there, this is familiar, you fall under the Clinical Laboratory Improvement Amendments Act of 1988, so CLIA. And these are for laboratories that perform wave testing or testing of moderate to high complexity. And it's regulated by the Center for Medicare and Medicaid Services, so the CMS. In contrast, the pharmaceutical side is regulated under the Federal Food, Drug, and Cosmetic Act. And this basically covers all pharmaceutical products, It covers medical devices, including in vitro diagnostic tests. It covers biotechnology products, so such as cell therapies, food and beverages, dietary supplements, as well as cosmetics for the US market. And this is regulated under the FDA. So where this matters is this intersection of health microbiology in this gray zone. So this gray zone has been expanding pretty slowly, but it's getting faster over the last decade or so, because this is where academia, tertiary care medical centers, and the pharma sectors are starting to intertwine, and where the two regulatory oversight bodies start to get, it starts to get a little bit confusing for end users that are in this gray zone. So I'm going to switch gears just slightly and talk a little bit about cellular therapies. So as you know, this is kind of the golden child of the medical age that we're in at the moment. But cell therapies have been around since the mid-1980s. Currently, there are over 5,600 cancer cell therapy trials that are ongoing globally, testing about 2,700 unique cell therapy products. CAR T cells seems to be hitting the lay media. So a lot of people will know the term CAR T cell. And that first one was approved by the FDA in 2017. And today there are more than 44 products that are approved by the FDA for cell and gene therapies, which is pretty amazing. Some of the CAR T cell therapies that have been approved are to treat ALL, diffuse large B-cell lymphoma, multiple myeloma. And then we also have approved gene therapies to treat sickle cell disease, leukemia, hemophilia, etc. So the expectation by the FDA is that the testing lab, so you want to make sure that these products are sterile. So it goes to a testing lab, should employ facilities and controls that are comparable to those used during aseptic filling operations or during manufacturing. And so here, this is another figure to emphasize that if there are academic clinical centers that are starting to perform phase 1 clinical trials on new types of cell therapies, there is an expectation by the FDA that current good manufacturing practices start to be employed. So we start to see emergence of clinical research with pharmaceutical expectations. And not only is there like joint commission oversight from a clinical standpoint and clear oversight from a clinical lab standpoint, we now have FDA expectations for GMP. So there are intersections, right? So these tertiary care academic centers that are now trying to do clinical trials are now also getting involved with needing to do sterility testing, which is a regulatory requirement for biological products. And it's basically to make sure that your product is free of microbial contamination prior to product release and prior to patient infusion. And there is a reason why many clinical microbiology labs or hospital laboratories are kind of getting pulled into this field. And it's because obviously the micro lab is close to the manufacturing suite. If you have a cell therapy manufacturing on your campus, it's much easier to walk down the hallway to the micro lab that's there. There's also on-site microbiological expertise. And of course, in-house testing is cheaper, it's faster, etc. And just to give the audience some context, this has been discussed for a number of years on the ASM listserv. So this is screenshots taken from ClinMicroNet of different emails that are posted to ClinMicro directors asking, we have a cell therapy lab that wants to be send settling plates and media fills to the clinical micro lab. I have a surgeon who wants to start an islet cell transplant program. They want me to do sterility testing. What are the regulations behind this? We currently use a blood culture analyzer for sterility testing. Is this done by Micro, or who else performs the testing? Someone else wants to start a bone marrow transplant program. They're being asked to do sterility checks. So hopefully that helps emphasize how the fields are starting to merge together and it gets a little bit gray. The other reason why clinical microlabs are also involved with sterility testing is that these are typically blood culture systems, right, which we use to look for blood culture infections as a sign of sepsis in a patient. But these have been shown to be suitable in the right circumstance for also product sterility testing. So if you already have a hospital lab that has these instruments and these types of media bottles, why not utilize them for cell therapy testing as well? All right, so let me put this into context of the NIH story. So essentially, the intramural program on the Bethesda campus at the NIH is renowned for discovering new drugs and new biologics for the last 40 years, and with a very, very long history. Typically, these are classified under investigational new drugs, and that's how they are claimed with by the FDA in the cell therapy trials. We manufacture on our campus cell, gene, and immunotherapies, viral vectors, PET radioactive drugs, small drug products and metabolites, as well as vaccines. And these are specifically for phase 1, phase 2 clinical trials in a bench to bedside clinical research setting. So the hospital that I'm talking to right now has patients literally down the hallway from me, but then also laboratories where they are manufacturing products that will eventually go back into those patients for under these clinical trials. So currently at the NIH, we have about 43 different products being manufactured across 19 different manufacturing facilities on our campus. So sterility testing is obviously very important. And then we knew this back in the 1980s, prior to my time, we knew that sterility testing needed to be done, and they employed the clinical micro lab to do this testing. In the early 2000s, we had a new clin micro director start in the hospital, and they showed that we could use those clinical blood culture bottles, and they were equivalent to the gold standard pharmaceutical sterility test. And they published some landmark studies at that time. And essentially, that is the testing method that went unseen without any director level oversight for the next 15 years. And in 2015, the entire program came to a halt because we had a contamination event in April of 2015 where a pharmacist was getting ready to release a vial of albumin, and they noticed that this vial had a mold ball growing in it. So of course they withheld it. They notified the proper authorities. They looked through the remaining 450 vials, and they found a second vial that was also contaminated with a mold ball. These mold happened to be Cladosporium and Aspergillus nigilans. This happened to lead to the FDA coming into the NIH hospital for a 4-cause inspection. They issued what is known as a Form 483, which is a list of observations from that inspection. They were on site for about, I believe it was 5 days and maybe a little more. And then the entire hospital pharmacy was shut down. So I guess I want to just pause for a moment and let that sink in. Imagine you are either a patient in a hospital, or if you're a clinical person and you're working in a hospital, imagine how a hospital can function without an active running pharmacy, let alone a research hospital, right? Which is focusing on all these types of different products. What is meaningful about this result is that the clinical micro lab, right, which is where sterility testing had been performed, signed out these products as being sterile and safe only 4 months prior. So while we were in the midst of responding to this, we had a second contamination event. So these were now a natural killer cell product that was coming down to the micro lab for final release testing, which happens to be a gram stain. And we all know that the sensitivity of gram stain is really not very good at all. So in this gram stain, although we happened to see septate hyphae, we went to the blood culture bottles that were incubating at the time, pulled them out of the machine, and saw gross mold contamination in them. There were multiple bottles that were positive with Aspergillus terreus. So this led to United States Congress mandating that a safety team be at the NIH for an entire year, and they released something called the Red Team Report, looking at and promoting patient safety. We were highlighted in very high-profile journals about safety lessons that we learned in our hospital, and we were on the front page of highly newspapers talking about the shakeup and what happened here. There was sweeping change of our entire hospital leadership in 2017. And we learned from the microbiology's perspective that clinical microbiology has no business doing GMP microbiology, which falls under the pharmaceutical sector. So ever since that time, I've been a strong advocate of trying to teach colleagues, especially within the academic setting. You know, if you're starting to get involved in clinical trials and need to get, you know, microbiology testing done, just be aware of what field you're stepping into. So essentially in 2018, this is where my career path changed. I went from being a clinical microbiologist into basically learning on the road GMP and pharmaceutical microbiology. We were able to build a brand new clean room suite dedicated solely for GMP microbiology. We stood up entire new programs, and our personnel have increased from 4 to 22 in the last 8 years. So here we go. If you happen to fall into this gray space, that is when FDA oversight takes over, regardless of whether you are doing this testing in a clinical laboratory. So my laboratory has been part of several inspections in the last few years. We've been part of 4 FDA audits. We mentioned the initial one in 2015 when they came through for the pharmacy. And that is at a time where the acronym of GMP, or good manufacturing practices, really didn't exist at the NIH. And we have done a lot of growth since then. I was then part of the FDA when they came through in 2016 for our cellular engineering group. They came back in 2019 to look at our hospital pharmacy, which included an unannounced inspection to the micro lab to look at our processes. And then again in 2021 with another cell therapy group. All right, so with that, let's move into the FDA inspection process. So the whole purpose of the GMPs, or the good manufacturing practices, it is all about control. We want to make sure that we identify critical areas of control to ensure the quality of the product. We want to make sure that we have identified what these minimum requirements are. We want to make sure that there are written procedures to describe how the control will be assured. We want to make sure that these written procedures are followed and documented. And then we want to make sure that when there are adverse events, that appropriate investigations have occurred to figure out how to remediate the cause of that out of specification result. So GMPs are used to create quality drugs, and essentially they are the minimum requirements required by the FDA for pharmaceuticals, biologics, and medical devices. They are intended to ensure that finished products are safe, effective, and in compliance with the Food, Drug, and Cosmetic Act. So this includes then safety, right? So does no harm, and that it is what it is supposed to be in its identity, that it is strength is sufficiently potent to deliver the therapeutic benefit, that it is pure, which means that it is free from contamination, and that it is quality and meets all aspects of GMP safety requirements. And basically, if there is a product that is being manufactured that does not fall within those requirements that I just mentioned for GMP, it means it's adulterated. And it means that those drug products and the persons that are responsible for manufacturing, testing, etc., and release of that product are subject to regulatory action. So the FDA inspection cycle can happen under 2 arms. It can either be unannounced, and they can just show up at your door if there is a for-cause inspection, much like they did for the hospital pharmacy here in 2015, or if there's like a whistleblower event, or there's customer complaints that are coming through to FDA, or they can be pre-announced. And if it's pre-announced, they'll issue a form 482. Essentially, the inspection would start with a tour of the facility, including the processes, products, records, equipment, and data sources. And then after that tour has been completed, the FDA inspectors will then begin their systematic inspection. So number 1, was it a for-cause inspection? They will obviously focus on the areas that they've been made aware of. and follow the cycle from there. If it is, whether it's a for-cause inspection or whether it's pre-announced, they will also follow up on whether there were previous observations that had been reported by the FDA, either listed in Form 483s or through verbal observations. And then they will also follow up on other regulatory topics and what happens to be hot at that particular time as what they're finding in other inspections from other facilities. They will then document the deficiencies and issue that 483, which is the list of observations. The facility then has 30 days to respond to that 483. So the FDA is specifically looking for evidence of compliance. They are looking for procedures. They are looking for written records and documents, with basically everything falling under the golden rule that if it wasn't documented, it didn't happen. So everything under the GMPs is a heavy focus on documentation. They may ask to review why decisions were made, so whether the corrective actions were made, operational changes were made, etc. And they will ask for supporting documentation and data to justify why those decisions were made at that time. All right, so they do it by following an investigations operations manual, which is, I think it was 800 pages when I opened it up earlier in the week. The inspectors will present identification. They have a badge that they'll present when they arrive. They may be either in plain clothes or in uniform if they are through the U.S. Commission Corps Public Health Service. And there is no difference in the way they dress and how they conduct the inspections. All right, so preparing for an FDA inspection. Essentially, a lot of this is basic knowledge, but it's kind of crazy how we need to, it helps to reinforce this through education. And this happens throughout my team as well, because you basically always need to be at the ready. You just never know when a regulator will turn up at your door. So essentially, to be prepared, just know your job and do it well. We should anticipate things. If there is a notification that they are coming in, we should anticipate what type of areas they may want to focus on. We should have documents prepared and have them readily available. We want to make sure that we have identified our subject matter experts within the team that are capable of answering knowledgeably directly to the FDA inspector. And we should have a plan strategy in place. So what is the flow? Who is responsible for what? So who is receiving the inspectors? What flow are they coming through the facility? Everything that they walk through, they may potentially take a look at and they may potentially take a photo of. So the flow path should be clearly planned prior to an inspection. Identify who the chaperones would be and who's going to be with the inspector at all times and how they depart the facility. The facility tour should be planned, who is giving the tour, again, the route of the tour and what is being said where during that tour, as well as any presentations that need to be prepared before time. So for example, for me, I usually prepare prior to an audit an overview of who my team is, what they do, what we are responsible for, how our quality system is set up, and it helps to give direction to the inspector on high-level quality management systems. In terms of the inspection setup, typically good practice is to have a front room and a back room. The front room is essentially where the inspectors sit. These are where the interviews will take place. You should identify who needs to be in the room at that time. Obviously, the subject matter experts need to be readily available, and someone with authority needs to be in the room at that time, as well as a scribe to take notes of the discussions that are going on. Then typically there is also a back room. This is where there is a lot of people who are working to gather all the data requests. And this is where the subject matter experts are prepared as well. So for example, an FDA inspector may ask for X number of documents or ask to see evidence of compliance with. And then essentially the folks in the front room, typically via some kind of system, we had a shared Excel document when we were doing it. and someone in the front room will be scribing what documents need to be pulled. The people in the back room will pull those documents and review them prior to them going in to the front room for review. The subject matter experts are also prepped in the back room. So it may be that notes are taken from the front room, shared with the back room, and someone can prepare them on what the ongoing discussion is, where the hang up seems to be, what kind of evidence they would like to see presented. And so the SME goes in prepared to that interview. The roles and responsibilities also need to be clearly delineated during an inspection. So we should identify who is the most responsible person in the room. And then if that person is not available, who is the backup to that most responsible person? And then from there, who is the backup for the backup? So just always have contingency plans in place. We should identify the host. So this would be someone who always stays with the FDA inspector, escort, runners, copiers, scribes. And copies of all materials is also kept by the facility themselves for whatever is handed over to FDA. Subject matter experts, obviously, as well as the tour guides. All right, so being prepared is all about quality system knowledge and having good team organization. So having your subject matter experts knowing what they need to know, having documentation readily available, reviewing the documentation in the back room prior to taking it to the front. The FDA does monitor response time. So don't think that because they ask for something in the morning and it's not available in the afternoon that they've forgotten. Do not make them ask for it again. And then essentially the data and the documentation just needs to be presented confidently, which means that it needs to be presented by someone who knows what they're talking about with regards to that subject. So the next few slides, I'm just going to go through a list of common mistakes that can sometimes occur during inspections. So essentially, never give false data. It is okay to say, I don't know, if you don't know the answer to that question, and try to find the subject matter expert who can answer that question confidently. Know that the FDA inspector may ask the same question to different people at different times. And for here, they are looking for consistency in the answers across the institution. Do not send the wrong person into the room because they won't be able to answer the questions correctly or confidently. I think this is in any type of audit or investigation or interview, the dome of silence, right? This is a key that anyone in management also is aware of, that silence is key. And sometimes it's done on purpose because naturally as humans, we feel very uncomfortable sitting in silence when there are other people around. And so sometimes information is offered when it wasn't asked. And so that is done on purpose. So essentially do not fill the space if there is silence. That is intended. Also, people involved in the investigation should not relax during the entire time. You need to be aware of what you are saying, what you are doing. You are always being reviewed and looked at. And this includes when you are outside the room. So if you go to the bathroom for a bathroom break, be careful what is said. During lunch, be careful what is said. There are ears and eyes everywhere during an inspection. There's also the factor. So the could be particularly from an investigator where they might just look at something and go, or it could be from someone presenting the data. It could be, that can be presented and perceived as problems. So it's encouraged to not shake your head, do not look concerned. All of those gestures and facial expressions are part of the investigation toolbox, and they'll present as a problem. If an inspector asks for information, data, or paperwork, do not make them ask for it again. They are very good at what they do. They have recorded what they have asked for, and they are monitoring the time that it takes to turn that around back to them. And just don't wait to see if they forget about the request, and don't make them ask for it again. Don't argue and don't challenge them. If you disagree, there's a way to politely disagree, but that would be countered with evidence, which is usually documentation of some sort. So do not stand around making faces or looking worried. Some of this is just general, standard practice essentially, but it alerts the investigator that something may be wrong. Be confident and proud in how the data is presented and how questions are answered, and do not be defensive of how that's presented. And if someone is being asked a question, they may not be the right person to answer, but if they are answering, don't contradict them in the moment. So do not answer for them, do not interrupt, and do not give contradictory information. That can all be sorted out later, but it just doesn't look good if there's a disagreement or an argument between the institution in front of an FDA inspector. Other common mistakes are interrupting the investigator or others while they're speaking, or speaking loudly when an investigator is trying to concentrate, read, or review documentation. They will ask for a lot of documentation and they will need dedicated time to look through this. Often that falls under the dome of silence. And so often you may be sitting there for a long time in silence and just being readily available to answer questions as they come up. Stick to the facts and not opinions. And listen to the questions really carefully. So clarify if you don't understand. If the question is too broad, ask for specifics, and then answer the question directly and do not give more information that's needed. So a good question that we always train our teams on is, for example, do you have the time? And the answer should be yes, or it should be no. It should not be, oh, yes, the time is 5 p.m. because then you've given more information than what was actually asked. I guess do not guess, for answers. Look them up in the procedure and present them as documentation to the inspector. If you do not know the answer and you are not the SME, make sure you get the right person in the room. And if you think the investigator has misunderstood an answer, make sure you try to correct that immediately and professionally. Show official records as evidence as well. All right. Communication is going to be key in any type of inspection. And I just pulled this data just to show that, verbal communication and what we say via speaking, only a very small percentage of that is actually registered in effectiveness. Close to 40% of communication is actually paralinguistic. So this has got to do with the tone, the volume, the timing, sounds, whether they're utterance or mutters that may come out. And then most of communication actually comes from non-verbal cues, which includes body language, gestures, eye contact, and your attitude. And often the non-verbal gestures are shown much earlier than the verbal response. And the inspectors are trained to look for these indicators when they ask questions. So in terms of documents that an investigator is permitted to review, they are allowed to see documents and records that pertain to control, manufacturing, or quality assurance within the facilities. These can include procedures and batch records, filings, QC procedures, validations, etc., and investigations. And they may also include training records and job descriptions, etc., to make sure that the right person is in the right job. Documents that cannot be reviewed by the FDA include reports of audits that are performed by the company themselves or by external auditors or consultants. So these are typically termed internal audit programs. They are not allowed to look at documents pertaining to financial matters of the company. They are not allowed to look at research and development data, and they are not allowed to look at personnel records other than training or management review meeting minutes. So what happens if something goes wrong during an investigation? We should discuss the concern or finding immediately, make sure that there is full understanding with all participating parties and that there are no misconceptions. And so questions need to be asked for clarity. At the end of each day, the company should ask for an oral list of all observations, which should have been recorded throughout the day as well. This is more of a means of summarizing the observations from that day. It also gives an opportunity for the vendor to address any of these issues prior to the close of inspection, and also to prepare any responses for potential 483 observations. At the end of the inspection, a list of these observations will be presented on a Form 483 in a closing meeting. And so you just want to make sure that the right people are in the room for this discussion. If the findings are minor, the investigator may opt to address this as a verbal advising only and may not include it in the official 483 form. And then the company has 30 days to respond to the 483, which must include corrective action plans to address specific observations and systemic corrections and timeframes for implementing these corrective actions. So just to give context, in the last fiscal year 25, the FDA conducted over 14,000 inspections. Most of these were domestic, with a small percentage being foreign inspections. For biologics, so these are cell therapies, for example, these accounted for only 9% of the total inspections. But obviously, drugs, food, cosmetics all fall within these 14,000 of inspections. The most cited areas from the FDA in the last fiscal year include procedures that are not in writing and are not being fully followed, the absence of written procedures. So again, if it's not written down, it doesn't exist. And inadequate investigations of discrepancies and failures. The top warning letter citations, so these are when these are beyond Form 483s. These are issued as warning letters to this particular institution. These citations include data integrity, the lack of process of validation, gaps in the cleaning and maintenance of their particular program, management of deviations, so when deviations off written procedures occur, and the lack of quality unit oversight. So again, you can see that a lot of these citations are focusing on quality and on control. All right, so this is my last slide. So essentially, the FDA is responsible for overseeing the entire pharmaceutical industry, the food industry, cosmetic industry, as well as biologics. But you can see that there is a creeping zone, right? There are some tertiary care medical centers that are dabbling in clinical trials of drugs and biologics, which are now moving the needle into FDA oversight of some clinical operations. The things I discussed in terms of preparing and behaviors during an FDA inspection, they're basically all standard behavior practices, but it's really key during an inspection cycle to be super aware and attentive of those behaviors and to offer those educational opportunities to staff. Quality system knowledge is going to be key, and written evidence is always going to be key, as they want this as documentation and the golden rule. And all this in the end, while it may seem like a ton of work and a lot of oversight and a lot of scrutiny, in the end is to make sure that manufacturers and testing labs are producing high quality products that meet specifications to ensure patient safety. So with that, I'm going to end. This is just a photo of my group last year. We're getting ready for another Christmas photo, I guess. But thank you guys very much for the invitation. I'm going to stop sharing, and I'm happy to take any questions that you may have.
Okay, thank you, Anna, for your great presentation. It was all very, very interesting. Can you all hear me, right? Did you hear me, Anna?
Yep, I can hear you.
Perfect. I myself, right? I used to work at the industry before I went into my PhD. So it was a really enriching experience. I learned something. I learned that I didn't want to be in industry myself. Not for anything wrong particularly, but I really love teaching. and I was missing that part in there for myself. So I was like wandering around, feeling uncompleted. But I really learned a lot, and I took a lot of it and applied into my courses. I even developed a GMP course for university. So it is really interesting what it is done in the industry and how and how it is extrapolated into research. So I want to open the floor if any one of the people connected want to ask any question. They can feel free to ask them via chat or you can turn on your microphone. So while they lose their shyness, I have a few questions I might ask. Uh, I wanted to ask, how is the FDA looking at the implementation of AI tools within the regulation
Oh, that's
system?
such a good question. Such a good question. They talked about this at the recent PDA FDA regulatory conference. They know that that AI is a big thing. In terms of the pharmaceutical sector, the FDA is not ready for this. They
Okay.
are it's too unregulated. They are aware of it. They are trying to build systems and programs in, but at the moment it is not an approved type of technology.
Okay, I ask in a way because I know there are certain companies already applying it right now, but they're mainly applying it for reporting, normalizing or standardizing the wording in the documents they provide. So it is all
And,
perfectly and
yeah, and maybe situational and maybe specific, right? I think it depends on how the AI is being utilized.
exactly.
And if it's being utilized as in a low risk model, that's
Mhm.
probably more acceptable than something that is more high risk, for which then they're going to be looking for the validation
For
behind that.
example, I just want to bring this example. If we're making a corrective action, preventive action method, right? For for how can I say that? Well, after the inspection, right? We have they highlighted something we want to correct. Uh, it is acceptable to do it with AI, for example, or they expect still the human person to develop the report.
My guess is that they're going to still rely on the human.
Perfect.
It would take a very robust validation to demonstrate to the FDA that all the gaps within the AI process have been fully closed. And I think
Perfect.
it's just such a fast moving field, right? And the technology is changing all the time that,
Okay.
in the end, they're always going to lean back on the human.
I think someone raised their hand. I don't know. I think I saw something, a notification around.
I
I don't
see
know.
some questions in the chat.
That's what I saw, the chat. Let me go there. Yeah, I saw that. Perfect. So I can read them for the benefit of everyone. It says that what is the difference between the aseptics procedure and the final sterilization for the products or medicine?
Yeah, it's a really, really great question. So sometimes there are drug products that can be terminally sterilized. So you can make them in a technically a non-clean room environment and validate your sterilization process that will essentially kill all viable microbes at the end and not affect your drug product at all. Right? There are those types of drugs. But then we have live biologics. So if you think about the cell therapies, right, there is no terminal sterilization because your
Mm-hmm.
cell's alive. And so that is when your aseptic practices need to be spot on. So often during GMP manufacturing, there will be multiple controls in place. There'll be environmental monitoring, there'll be glove fingertip testing, there'll be settling plates, there will be many, many controls within the process to ensure the validity of your result at the end.
Exactly.
So I guess that's the answer to that, right? You can be a little bit more uh, le- a little bit more like careless if you have a final sterilization, as long as that's been validated and you can prove that it can truly kill whatever bioburden you have in there at a particular time versus a live therapeutic. Same thing goes with probiotics.
Also, I have another question from Maria Santiago. It says, I am new working in food industry. Many of the points discussed today also apply for inspections in my area, right?
Yes, absolutely.
Yes, okay. There are any other additional questions from the people in the in the talk? So I have one last question. I wanted to ask, how do you see your field going moving forward in the next years with all these new technologies coming up, and not just coming up, they're coming up fast, and just revolving everything around them, and people just want everything now more faster, quicker because of these tools. So how do you see your field with sterility? It is a slow procedure by itself, by nature. So how can we handle the new culture of I want it now, I want it fast?
Yeah,
So,
yeah. I'm on board with you. So I'm on the USP micro expert committee, right? Some of these methods that we use, so molecular diagnostics and even the blood culture systems, right? They've been around forever in other fields, right? Clinical micro, and in other fields, maybe in food industry and stuff. The pharmaceutical side is very slow to adapt to new methods. And that is because it's generally risk adverse, right? You need to have just so much data across so many different types of products. What you manufacture may be just slightly different to what I manufacture. And so it's really hard to apply a technology broadly in that sense, as opposed to a patient specimen, right? Where blood is blood, sputum is sputum. The USP Micro Expert Committee is very much aware of this. We just released 2 chapters on rapid testing. One includes the blood culture system. The other one includes ATP, bioluminescence, for sterility testing. We are working on a molecular chapter for mycoplasma testing. That should be hopefully going out next year. But again, we're essentially 20 years behind everyone else. And again, I think it's just because it's risk adverse and the amount of data needed to support a regulatory chapter of that technology.
Yeah, I believe that most companies, they believe that if it is already working, why change it? We already invested a lot of money into validating these procedures as they are.
Exactly.
They are
Yep.
working and we are making money out of it, and we're paying our investors, which is a big part of companies. So I think I see why they don't do it, right? So yeah,
I think
thank
to add to that too, a lot of pharmaceutical companies are global. So you don't want to be doing a different testing method in the US versus what you're doing in Europe and Asia and Australia, whatever, right? So it's usually a global change that happens, which means that the regulatory guidance needs to be similar across the EP, the European pharmacopoeia, the US pharmacopoeia, the Japanese pharmacopoeia, etc.
I think they call that they have like a harmonization effort.
Harmonized chapters.
Yeah,
Yeah.
yeah. So that's very, very important. There are any other additional questions within our person connected? I don't see any other questions in the chat. There anything else that you might want to add to the
Hi,
talk?
excuse me.
Antonio, welcome. Thank you.
Yeah, I'm a student of subgraduate in the University of Puerto Rico. I want to ask, what are the biggest challenges when you are making a sterilization test?
The biggest challenges are having your controls in place and minimizing contamination. I mean, the results that my lab puts out are either the go or no go for whether a patient receives their therapy or not. So I can give you guys, a real case study. We had a CAR T cell at the end of October that came up positive on day 10 of incubation with gram-negative rods. And it ended up being identified as methylobacterium, right? So a strange organism, an environmental organism, a slow grower. It doesn't grow typically. There was a risk. The patient was being flown in to the hospital. They were scheduled to start their lymphodepletion to receive their therapy 5 days later. But now we have this positive sterility result come up. We have a risk assessment committee, which includes, the clinical team, me, the manufacturers, etc., like all the stakeholders involved. And the decision is made not to infuse the product, right? But you have a patient who's like, has failed all other therapies, right? And they're here because they're in a first in human trial at the NIH. So then, as part of the investigation, the manufacturer sends retention vials, right? So these are secondary vials that are essentially frozen as a backup just in case. So they sent those to me in beginning of November. And today, the retention vial of the same product came up positive for the same bug as day 10 of incubation. It's the same organism. And so that could have been a near miss event, right? We really are that stopgate as to whether a patient event happens or not. And this happens all the time, right? I'm just telling you one event that has to be very recent in my head. But you can imagine just across the field of pharmaceuticals, food, cosmetics, right? That is why that control needs to be in place. And so to answer your question, Antonio, like the most challenging thing is making sure that you can trust your results. And with that comes all the controls, all the validation, all the training, etc. Because a positive result in good practices, right? A positive result should happen very, very rarely, which means that when it happens, it's real and you believe it and you make the right clinical decisions beyond that.
Thank you for the time.
There are any other additional questions in the public? Which method for identifying those contaminants you use there at NIH? Do you use MALDI-TOF or do you?
Yeah, we use MALDI, and then we reflex the sequencing if it doesn't ID by MALDI.
Okay. So always the sequencing will be the gold standard, probably Sanger, right?
Yeah.
Okay. So if there are not any other questions, I think we already reached the time limit. I think it's until 6, right? So I'm really grateful for you accepting our invitation today, Dr. Lau. It was really insightful, your presentation. I think we'll all learn a lot. And we're really grateful for your time. And we hope to see you again in other activities around with us in the Puerto Rican chapter of the American Society of Microbiologists.
Happy to. Thank you so much for the invitation. Thanks everyone for joining.
See you later. Bye-bye.
Bye-bye.